Today's presentation is Submerge and the Oligonucleotide Emerge Best Practices for Developing These Complex Biologics. Our speakers are Dr. Martin Billibo, Dr. Suzanne Minton, and Dr. Verna Schuck. Martin is currently a Senior Director at Certara IDD. He holds a doctorate in public health from the University of Montreal. Suzanne is a Director of Content Strategy. She earned her doctorate in pharmacology from UNC Chapel Hill. Verna is Senior Director at Certara IDD. She holds a doctorate in pharmaceutical sciences from the University of Florida. I'll now turn it over to Suzanne to begin the presentation.
All right, thank you so much, Pam, and welcome everybody to the webinar. Just trying to get, there we go, control the slides and advance it to the next slide. Being a little fiddly, there we go, ooh, okay. All right, so today's presentation, you can think of it as a play in 3 acts. And so in Act 1, I'll be talking about oligonucleotide therapeutics and how they work. In Act 2, in Act 2, Martin will be talking about the preclinical and translational considerations that oligonucleotide drug developers need to be aware of. And then in Act 3, our colleague Verna will be talking about clinical pharmacology considerations that you need to be aware of. So in getting ready for this presentation, I did a little bit of market research and a couple things stood out to me. First of all, this market is exploding. There's at least 135 oligonucleotide therapies currently in development. Moreover, in terms of the business opportunity, this is a rapidly growing market. It's been estimated that by the year 2024, the global nucleic acid therapeutics market will reach more than $7 billion. And finally, I went to PubMed just to look at the publications in this space. And if you enter the term oligonucleotide therapeutics, you get in excess of 35,000 hits with a dramatic increase in the number of publications in recent decades. So what is the history of the development of this therapeutic class? So nucleic acids, DNA and RNA, were first discovered in 1868. But the vast majority of the basic research that went into forming the foundation of this therapeutic class was done in the 20th century. I'm sure we're all familiar with the central dogma of DNA, or central dogma of biology. This is the idea that DNA is transcribed to RNA, which is further translated to protein. So scientists realized that if we can create oligonucleotides that interfere with the expression of RNA that's linked to disease-causing proteins, that these entities can then serve as therapeutics. So in this timeline, it's color-coded, and you can see that the milestones in biology are teal-colored, which later support milestones in chemistry, which are marked in green, which later culminate in clinical milestones in dark blue. Scientists first came across the idea of the antisense concept. That is the idea of using a synthetic antisense oligonucleotide to control gene expression. 20 years later, the FDA gave the first antisense approval to formivirsin. Likewise, in 2001, came the discovery of RNA interference mechanism. And this led to, in 2018, the eventual approval of patisiran, the first siRNA, small interfering RNA therapeutic. So how do oligos compare with drug development issues that one might face for small molecules and biologics? So I don't think I need to tell this crowd all that much about small molecules drugs. I'm sure you're all very familiar with it. In this presentation, we'll be focusing on a couple different types of oligos. There are the antisense oligonucleotides, which are single-stranded DNA snippets. There's also the siRNA, which is double-stranded RNA molecules. And then drug developers sometimes conjugate these oligonucleotides to ligands to help increase the targeting to specific tissues. And so the best known example of this is GalNAc, which is a ligand that helps target oligonucleotides to hepatocytes, because they strongly express the receptor ASGPR, which is the receptor for the GalNAc ligand. And there are other types of oligonucleotide therapies like synthetic messenger RNA and aptamers, but they're beyond the scope of this talk. So let's compare and contrast some of these classes. So in terms of size, small molecules are the smallest, and then oligonucleotides are actually intermediate in size. They're bigger than small molecules, but they're smaller than biologics like peptides and proteins. In terms of their physical chemical properties, they're highly soluble, and because they have that sugar phosphate backbone, they're highly negatively charged. They're polyanionic. In terms of their pharmacokinetic characteristics, these oligos are highly distributed to the liver, the kidney, fat, lymph nodes, spleen, and bone marrow. Notice I didn't say the brain. This is actually important for CNS drug developers because oligonucleotides tend to be too large and also too negatively charged to cross the blood-brain barrier. From a therapeutic standpoint, this means that if you're looking to target a CNS indication, the oligonucleotides have traditionally had to be administered through more invasive methods like intrathecal injection, where the oligonucleotides are directly introduced to the cerebrospinal fluid. Oligonucleotides are eliminated by the liver and the kidney. And then how are these drugs administered? Small molecule drugs have the benefit of often being able to be administered orally. By contrast, oligonucleotides and biologics have historically been restricted to subcutaneous and intravenous administration. I do have a little asterisk in here about oral administration. Some recent research out of AstraZeneca, which was presented last month at the DMDG meeting by Genemark, talked about how you can use absorption enhancers such as sodium caprate to enhance the intestinal permeability of oligonucleotides to facilitate oral administration. And it seems pretty clear how this could be of benefit to patients. And then how are oligonucleotides broken down? Their metabolism is chiefly mediated by nucleases. And then lastly, what do you have to consider with toxicity? So one of the issues with small molecule drug development is off-target toxicity is often an issue. However, oligonucleotides are rationally designed against their mRNA targets, and so off-target toxicity doesn't tend to be as much of an issue. Immunogenicity is the issue of administering a drug and then having the patient's immune system mount an immune response to it, and this is a well-known issue with biologics. Likewise, antisense oligonucleotides represent novel structures that have a potential for immunogenicity. So this is something that drug developers should be aware of. And what is the mechanism of action by which oligonucleotide drugs work? So again, this presentation, we're going to be restricted to antisense oligonucleotides, siRNA therapeutics, and conjugated versions of these oligonucleotides. So both antisense oligonucleotides and siRNA work by mediating degradation of the target mRNA, which blocks expression of the protein target, but they do it in different mechanisms. So antisense oligonucleotides bind their target mRNA through Watson-Crick base pairing, and then they mediate degradation of the mRNA through RNA ACE H1. siRNA, remember, it's a double-stranded DNA entity. It enters cells and forms a complex with a group of proteins called RISC. These unwind the double-stranded complex, and then the siRNA strand that's complementary to its mRNA target, it binds it and then mediates degradation of the mRNA. And then lastly, I'd like to talk a little bit about how these entities are formulated. So most antisense oligonucleotides can be administered in simple formulations like saline, but siRNAs and modified mRNAs generally require a protective formulation. And so this is most commonly done through an LNP, a lipid nanoparticle delivery system. And so you can see here in this cross section of this micrograph, you can see what this looks like. So you see in red the double-stranded siRNA, which, remember, it's negatively charged, and it's bound electrostatically to positively charged cationic lipids. And so you can see that there's a mix of lipids here and that they form a particle, and that this is the way that these therapeutics are typically administered. And so with that, I'd like to thank you for your attention, and now I'm going to pass it on to my colleague, Martin.
Thanks, Suzanne. Hi, everybody. So the second act of our presentation is going to be tips and tricks, I would say, about translating animal to human information on oligos. Well, some examples of recent FDA-approved oligos, a lot of it is focused on rarer diseases. For example, here we have spinal muscular atrophy, we have familial hypercholesterolemia. But there's a lot of antisense oligo work that's going on in the field of viral suppression as well. And in fact, this work has been considered pioneering work regarding the cross-species extrapolations, where a lot of the developments were made. In this space here, recent active player in the field are Replicor and Aligos. What are the oligos? What are some of the pharmacokinetic characteristics of the oligos that we often develop? Well, their PK is often governed by their type of modification. For example, ASOs that contain a phosphorothioate backbone, they're extensively bound to plasma protein. We're talking about more than 85%. And that protein is mainly albumin. That's common across all species, including humans. This high degree of binding to this plasma protein, it reduces renal excretion and renal clearance of the drug because it doesn't go through glomerular filtration. That binding affinity to albumin is relatively low. That allows uptake to the tissue. The siRNA drugs are less charged or less bound to plasma proteins, for example, siRNAs, and they are cleared more rapidly from blood because because of either some nucleus metabolism in blood or excretion in urine. For example, less than 2.1% of partisiran is bound to plasma proteins, and the terminal half-life of elimination for this drug is only 3 days. The other effect or the other impact that some of these modifications would have is their conjugates. For example, if you compare ADCs versus GalNAc, such as patisiran and inclisiran, these single chemical entities are targeted to the liver, and they allow for some easier subcutaneous administrations. The other impact will be what the carrier of this RNA is, like Suzanne just mentioned, the NLPs for partisiran, for example. These are lipid formulations. An LNP is around 85 nanomolars. Again, they allow targeted delivery in liver, and usually when they have such a carrier instead of a conjugate, they will be given by an IV administration. Let's talk about how you can leverage some of the early preclinical information to inform your clinical development program. And we'll separate the talk into 3 main categories, and those of you that are familiar with pharmacokinetics will recognize the categories here: category of absorption, distribution, and metabolism and elimination. What are some of the expectations when we talk about absorption? The primary route of absorptions for oligos for systemic application is by parenteral injection, either intravenous or infusion, subcutaneous injections. Following systemic administration, the modified single-stranded ASOs, they rapidly transfer from blood into tissue, and that can take minutes to hours. The pharmacokinetic properties of ASOs are similar across species and gender. The rapid transmission into cell is predominantly facilitated by endocytotic uptake. Once intracellular, ASOs exhibit long half-lives and prolonged activity in suppressing or altering the target RNA. Following subcutaneous administration, the ASOs are rapidly absorbed from the injection site into circulation, with peak plasma concentrations consistently reached within 3 to 4 hours. Nearly complete absolute bioavailability has been observed for multiple ASOs after subcutaneous administration, at least in monkeys. Following either IV or subcutaneous administration, plasma concentrations rapidly decline from peak concentrations in a typical multi-exponential fashion. This is characterized by a dominant initial rapid distribution phase, where the drug transfers from circulation into tissues in minutes or hours, followed by a much slower terminal elimination phase, where the half-life can go up to several weeks. The apparent terminal elimination rate observed in plasma is consistent with the slow elimination of ASOs from tissue. This indicates equilibrium between the post-distribution phase plasma concentration and the tissue concentrations. What happens when you have some incomplete sampling profiles between your subcutaneous and IV terminal phases? Here, note the absence of a post-distribution phase following your subcutaneous administration, where you see a typical mono-exponential decline, whereas you see a multi-exponential decline following an IV infusion. And this may be one of the reasons why you would see this. Did you sample long enough following IV infusion and subcutaneous administration? You're supposed to see a post-distribution that's 100 hours plus. The problem, if you're not capturing the multi-exponential decline, is you can potentially scale the flip-flop kinetics that are happening. Flip-flop means, or is the phenomenon, whereas you would predict or extrapolate to humans the absorption rate instead of the elimination half-life. What's the solution to a situation where you have such incomplete or discrepancy, you have different information following your IV or your subcutaneous administration? One solution is to use modeling and simulation to simultaneously describe the plasma, intravenous, and subcutaneous administration. This allows you to leverage the strength of each data set. In this case, you could have the pre-systemic absorption compartment, that mono-exponential phase after subcutaneous data, inform the initial pre-systemic disposition, and then you can have the IV data, where you see in the prior slide a more slower half-life, or this two-phase distribution, inform the systemic elimination of the drug. Let's move on to distribution once the drug has been absorbed. What are some of the information we can get from that? Well, it's fairly consistent across species that the partition ratio between the liver and the post-distribution plasma concentrations are similar. And it's about 5,000 to 1 for a lot of these modified ASOs. Therefore, the post-distribution plasma concentrations are expected to provide a surrogate for tissue exposure in humans. The ASOs that contain this phosphorothioate backbone are extensively bound to plasma protein, as I just mentioned, and albumin, again, is the biggest culprit in this drug binding. The protein binding to albumin is relatively low affinity. That prevents the loss of the drug to renal filtration, yet it still facilitates uptake to tissues. The plasma protein binding is not saturated at clinically relevant doses because of the large capacity of binding to proteins in plasma. In contrast, the oligos that lack a charge or that less extensively or more weakly bound to plasma protein, such as peptide nucleic acids or unmodified sRNAs, they exhibit more rapid clearance from blood, primarily due to either metabolism in blood or excretion in urine. These compounds and their metabolites are readily filtered and excreted, resulting in much lower or negligible tissue uptake. For these reasons, the kidney, the organ with the highest concentrations of these oligos, is the primary organ of distribution, but it's still very low concentrations. Thus, an appropriate and balanced plasma protein binding profile is required for broad and significant delivery to tissues and cells systemically. The ASOs are if the ASOs are either too tightly bound or not bound enough to plasma protein, that will result in poor distribution properties. For all animal species evaluated, ASOs distribute broadly into most tissues, with the exception of the central nervous system after systemic administration. The major systemic tissues for distribution, they include liver, kidney, bone marrow, adipocytes, the cell body, but not the lipid fraction, and the lymph nodes. So what happens when we have different post-distribution phase profiles between our plasma and tissue? Here we have an example of a BQL effect, where the sampling in plasma up to 24 hours above BQL, but after 24 hours drops below BQL. We therefore don't have this long post-distribution equilibrium phase appearing in our data. Yet when we analyze liver and kidney, it's fairly consistent with what we see in the literature, this long half-life that's similar across tissues. And that's certainly much longer than the half-life that we see in plasma. So if we were to take our value of 10,000 and divide it by 5,000, which is our plasma partition ratio, we would get about 2 nanograms per ml in plasma. So no wonder we don't see it. It just falls below our detection limit. What do we do in these situations to reconcile these different kinetic profiles between plasma and liver? Well, it turns out the answer, once again, is to use modeling and simulations to simultaneously describe the plasma and the tissue. Again, that allows you to leverage the strength in each data set. You can have the pre-systemic data, like we saw before, inform the absorption rate and the bioavailability of the drug. You can have the IV data inform the systemic distribution, and you can have the tissue data inform the slow, long terminal half-life. So each component of each data set contributes to the overall picture of the plasma distribution and the tissue distribution kinetics. What about metabolism and elimination? How can we use some of that preclinical information when we consider our clinical development program? Well, turns out it's fairly easy. The post-distribution profile scales approximately by body weight from monkeys to humans. And you see that in this table that I presented here for a series of ASOs that shows that the monkey and human C trough are fairly consistent. They're fairly equivalent when you scale the drug on a milligram per kilogram basis. And the reason for this is the consistent, predictable clearance across species. The clearance increases directly proportional to body weight across all of these species. So if we do some allometric scaling, we get similar plasma distribution phases and kinetics across sequences and between species, except the mice. You see here the mice is a bit of an outlier in this graph. And in general, in fact, rodents are more sensitive to class-related pro-inflammatory effects of ASOs compared to primates. That includes humans, and therefore they're not considered an appropriate species for the calculation of systemic safety margins. So not only is there a kinetic reason why they don't behave the same, their clearance is a bit of an outlier. There's also a relationship to toxic events where they're more sensitive. Therefore, they're not generally considered a good species. As seen in the previous slide, the pre-K scales by body weight from monkeys to humans. And that's generally held as a rule of thumb, but it's not always a perfect match. So if we take some individual compounds here, and I've put some human AUCs and human Cmaxes, and if you were to do the allometric scaling exercise, you find that yes, sometimes it scales directly to body weight, like when this ratio here equals 1, but sometimes it's a bit lower or a bit higher depending on the ASO of interest. So yes, it's a good general rule of thumb, but it's important to use all of the information in the preclinical species that you have to be able to verify whether that's the case or whether you have a bit of an outlier that might be a little bit lower and doesn't really scale perfectly to body weight. Understanding how ASOs traffic into and within cells has the potential to improve ASO design and ultimately lead to more potent ASO drugs. In vivo experiments in mice demonstrated that slowly infused drugs systemically in vivo result in a substantially greater uptake in the liver compared with bolus administrations, implicating a saturable uptake process. The lower plasma ASO concentrations presented to the liver over a longer period of time via infusion allowed a larger amount to be taken into cells. However, there was little to no ASO activity associated with the higher concentrations in liver, suggesting that a non-productive uptake pathway exists in cells that endocytose ASOs presented at low concentrations. Thus, better productive uptake in vivo, for example in liver, was facilitated by bolus injections of the ASO. That's because it could overcome the saturable non-productive pathway. If we use some nonsense ASOs to compete with this non-productive pathway, we get less bulk uptake in liver and pretty paradoxically, greater antisense activity as measured by the target RNA silencing, even though we have lower total exposure. So this suggests at least two pathways of uptake in cells in vitro and in vivo, one leading to productive antisense activity and the other which results in sequestration, non-productive uptake of the ASO. Early endosome uptake and sorting pathways may explain this. Further, intracellular binding may be associated with some localization or trafficking of the ASO. The ASO uptake in these cells implicate endocytosis as a dominant uptake mechanism requiring binding to surface proteins. Further, the importance of DNA in the productive uptake of oligos was confirmed, where both the base and the deoxy sugar were required for optimal uptake. Nanomolar concentrations of ASO taken up by endocytosis reduce target RNAs within hours and maximum knockdown within 24 hours, which is similar to in vivo observations. Microscopy imaging demonstrated that the bulk ASO found in the cell is immediately and predominantly associated with intracellular vascular structures consistent with lysosomes. Recent progress in identification and utilization of ligand conjugation strategies confirm that potency can be substantially improved through more efficient uptake in the target cells. GalNAc conjugates have greatly enhanced the target distribution of these molecules to hepatocytes. In general, liver, and specifically hepatocyte distribution and activity is enhanced approximately tenfold when concomitant reductions in distribution to other organs and other cell types. Increased potency of these molecules as manifested by reduction of target mRNA at lower and lower doses is directly correlated with the increase in exposure to the hepatocyte. While this specific ligand appears limited to hepatocyte targeting, it nevertheless points the way or paves the way to potential optimization for other cell types to cell-specific endocytotic pathways such as specific antibody drug conjugates. what are some of the, what is the third pitfall that we see when we're talking about metabolism and elimination, is assuming that the half-life of the ASO and the effect are directly related. That's just not the case. The time course for PK sampling of the sRNA can be shorter than the ASO, but the time course of the PD sampling can be longer. This is because the total elimination half-life of the sRNA is shorter than the half-life of the ASO due to the distribution of ASO in tissue and the slower clearance from the tissue. Often, though, sRNA therapeutics have short elimination half-life. The dosing frequency can be up to 6 months, therefore, and the dosing frequency is weekly or monthly for ASO drugs. Even though the clearance is very fast, you can get away with dosing it less frequently because of the long terminal half-life and the accumulation in tissue. There's also a temporal disconnect between the plasma PK of oligos and their prolonged PD effect. Choosing the dosing interval and making dosing adjustment through the length of the drug effect and the PK covariate is key. You do not use the duration of drug exposure. For example, inclisiran is eliminated in about 48 hours, but you still use a 6 months dosing interval that's in the phase 3 study and in the label. So what is the solution to this pitfall of disconnect between the pharmacokinetics that you see in plasma and the pharmacodynamic effect that you see that can be much longer? Again, try to use modeling and simulation to simultaneously describe the PK and the PD. We saw our model gets more and more complex. We start with some IV data, we add some subcutaneous data to leverage the information in each data set. Now we're adding the pharmacodynamic data and the tissue data to be able to describe the distribution to the liver, the binding to the HSPR receptor on the hepatocyte, the internalization, the escape from the endosome, the cytoplasmic risk binding, the degradation of target RNA, and the inhibition of the disease-causing protein. There are many intermediary steps that are nonlinear and time-dependent. That complicates the dose-exposure relationship for these drugs. And you can either go very, very complicated with a quantitative system pharmacology model, or you can try to simplify the response as much as you can by directly relating the dose to the response or the PK in tissue and linking that tissue pharmacokinetics to the response. The exact framework and the exact methodology and model you will use will depend on the data you have available on hand and the objective of what you're trying to do with the model in the clinical setting. In summary, how can preclinical information inform the clinical pharmacokinetics? In terms of absorption, beware the flip-flop. You have some possible terminal phase bias. It can appear faster than what it actually is. And to be able to model that correctly or account for that correctly, you need to decouple the absorption component, the bioavailability, to the clearance component. And remember that your clearance component is the one that's going to scale directly to body weight across preclinical species. Second of all, in terms of distribution, remember that your drug will rapidly, extensively distribute to tissue after dosing. Your plasma post-distribution PK will scale by body weight from monkey to humans, and your tissue concentration between monkey and humans will be similar on a milligram per kilogram basis. Again, because of that linear proportional relationship between body weight and clearance across preclinical species. In terms of metabolism and elimination, really we have predictable clearance. Our clearance increases directly proportional to body weight, but you have to remember the non-linearities which might be present when the ASO gets transferred to the tissue. There might be some saturation kinetics happening during the mechanism of cellular uptake. There's often a temporal disconnect between your plasma PK of your oligo and their prolonged PD effect. And in all of these situations, a good model is often key in understanding how all of this interrelationship interacts. Now I will pass the presentation to my colleague Verna.
All right, great. Thanks, Martin. Can you move on to the next slide?
There's usually just a little bit of a delay. If you try clicking it, there you go.
There you
You got
go.
it.
Okay, thank you. All right, so what I'll cover next are some clinical pharmacologist considerations when developing oligonucleotides. And the intent here is just to give a couple of an overview and a few examples, of important differences or important studies that we should consider in the development of this type of molecules. As Suzanne covered initially with her talk, there are different classes of oligonucleotides that are being developed as therapeutic agents. Because these molecules play a significant role in regulating the biological process with the intent of treating the disease, they end up having a very prolonged pharmacological effect. All these unique characteristics of oligonucleotides and the limited knowledge that we have when it comes to clinical data, PK, safety and efficacy, then makes the development of this type of molecules more challenging. I think we are evolving in the clinical pharmacology field in terms of how much we understand for this type of compounds, and we are building sort of a database, if you wish, that can help us guide the development of any new molecule that comes from this class, from making chemical modifications or conjugations to treat different disease. So when developing oligonucleotides, I think it's important that you consider all aspects of clinical pharmacology, that you understand the PK properties of your drug and what can impact exposures to better define and optimize the doses for the patients. What I'll cover next in this presentation is essentially go over a bit of the pharmacokinetics properties with some examples, the drug-drug interaction data, and then renal and hepatic impairment. As Martin covered during his presentation, the PK of oligonucleotides are primarily a function of their backbone and chemistry. Modifications to that backbone and conjugations to it have resulted in changes in tissue binding, protein binding, and bioavailability or half-life. One example of that is presented in the figure on the right, where you see the top three curves are the profile of a parent ASO, and the bottom three curves is the profile of the same ASO now with a GalNAc conjugation. As Martin explained, certain conjugations then target specific tissues and increase distribution into the tissue. And that results in then you having a lower plasma exposures, which is being demonstrated here in that figure. But overall, elimination half-life of these two agents is very similar and comparable, but there are differences, in the plasma exposures and the tissue distribution. These aspects are important to be investigated in a non-clinical setting, so you can look at the absorption, distribution, elimination, etc. And specifically when it comes to tissue distribution, those are important information to collect in the non-clinical space. There has been certain consistency in terms of the distribution across different tissues and across different animals that makes the extrapolation from the non-clinical information to the clinical setting a little bit easier. The reason why considering running or having this information in non-clinical species is to support your clinical setting or what experiments you want to perform or your studies that you need to perform when going into humans. But also radiolabel studies with oligonucleotides are usually not feasible and not required because of that long tissue half-life that the compound has, and makes this type of studies really challenging or difficult. But that being said, these are still things you need to explore in your phase 1 studies. In terms of a clinical pharmacokinetics, I mean, oligonucleotides are usually dosed as IV or sub-Q, but there are efforts in developing oral formulations that would allow oligonucleotides to be taken orally and make it more comfortable for the patient. The clinical profiles are very comparable, similar to the non-clinical profile as Martin described. You see fast absorption after sub-Q administration. There seems to be a flip-flop kinetics with that plateauing of exposures until you see a clear elimination phase. In general, you see dose proportional pharmacokinetics at the clinical doses, although some non-clinical more than dose proportional exposures may be observed. If you see saturation, depending on the type of oligonucleotide you're working with, there may be saturation in the distribution into the tissues if your target is specific receptors, that in consequence will result in having more drug in the plasma. Because of the long-lasting pharmacological effects that you see with some of these drugs, the dosing is usually sparse within weeks or sometimes months, and therefore there's no PK accumulation that is seen. Demographic parameters like age, gender, race do not impact the PK properties of these compounds, although some correlation has been seen with body weight. And some of these agents are dosed in a mg per kg basis, while some other oligonucleotides are dosed as a flat dose in milligrams. The conjugation can impact the distribution of oligonucleotides, as Martin described or discussed previously. The plot you see here is an example from givosiran, which is a GalNAc conjugated ASO. The solid lines represent the parent GalNAc drug, and the dashed lines you see there are the main metabolite that were observed in plasma samples, which is formed based on the loss of one nucleotide from the 3' end of the molecule. In general, the parent is the main, the major component or the main circulating species that you see in plasma, and these metabolites tend to be in a very smaller proportion or percentage relative to the parent. But in this case, the main metabolite for givosiran was actually present in a good amount, and it was about 35 to 75% of the parent across the different dose ranges that were investigated. So it is important that during the phase 1, you look into doing some investigation in terms of the parent, the presence of the metabolite, and how the magnitude of how much metabolite is present relative to parents. Some of these metabolites can be active and can contribute to the pharmacology. So understanding the amount of the presence is important in plasma, as well as looking at urine, because this molecules are excreted in urine. And then understanding the contribution of urine clearance is important to help you decide later on if a renal impairment study is necessary. In terms of drug-drug interactions, there is a low risk of drug-drug interactions with this class. Usually, oligonucleotides are metabolized by endo or exonucleases. There's no involvement of CYP450 enzymes in the metabolism of these drugs, and they're just, as I explained, the endonucleases would just cleave a nucleotide out of the ends of the oligonucleotide, the 3' or the 5' end, and make shorter length fragments, while the endonucleases may cleave the molecule in more inside in the middle of the structure. But because these are large molecules, negatively charged, hydrophilic, they do not typically interact with CYP enzymes. They have not been demonstrated to be substrates, inhibitors, or inducers. It's the same thing with transporters. That being said, there is a possibility of drug-drug interactions from a mechanistic perspective, which can be related to the pharmacological effects, and I have one example on that. that can present later, and there's also a possibility of indirect DDI due to cytokine storms. So knowing how your drug behaves in vivo is important for you to decide what studies you need. So you need to look on a case by case and make that case whether you need to run a DDI study or not. There is no specific regulatory guidance on DDI, so the examples are shown next. It's essentially applying the current DDI guidance to decide whether clinical DDI is needed or not. In most cases, the clinical DDI was investigated either in dedicated studies or via population PK analysis. For example, if there are common co-administered drugs in the patient population, you can explore drug-drug interactions with a common co-dose drugs in population PK approach. So far, from the drugs that have been approved, all of them included some DDI assessments in vitro or in vivo, but there has been no specific dose change or recommendation of any dose change due to drug-drug interaction. In general, I think the in vitro studies for DDI is very simple and straightforward to run, and there is recommended that you do collect that information to support your submission. From victim DDI perspective, there is little to no risk. I think many results or studies have demonstrated that these are not substrates of CYP450. There is a publication where they investigated in vitro DDI for different GalNAc sRNAs and did not show any involvement of CYPs in the metabolism of different type of compounds, which is presented in the table here on the slide. There may be a little bit of contribution of some of these enzymes, which is a very low percentage when it comes to just end the methylation or all the methylation of the link for the conjugated GalNAc sRNAs. But overall, no significant contribution. And in my experience, we actually helped one of these molecules with the submission for one of these molecules, which did not specifically include a CYP phenotyping assay, but we are able to actually build a case based on the knowledge of the metabolism of these drugs and based on the knowledge of similar oligonucleotide classes or conjugates, that the data was not needed. From a perpetrator DDI risk, there's also data suggesting that oligonucleotides are unlikely to have any perpetrator DDI effect. As I mentioned, the past approvals at the FDA of this class of molecules, they all included some in vitro perpetrator assessments, but there have been no in vitro induction or inhibition of CYP enzymes that has been reported that are of clinical significance. The same publication that investigated the different GalNAc conjugates also look as substrates, also look at them as inhibitor of the enzymes, and there are a couple of these compounds that actually had a weak inhibition of 2B6 and 2C8 as presented on the table here. The authors then suggested that the results of the in vitro DDI inhibition then be assessed in terms of the risk of a clinical DDI by just performing the assessments proposed or suggested in the FDA and the EMA guidances in looking at the in vitro KI inhibition relative to the in vivo Cmax. In this case, the maximum concentration was actually, they looked at the plasma as a first step, but they also considered the liver concentrations. Because now GalNAc conjugates, they target the liver, they tend to have liver concentrations that are multiple fold higher than the plasma. So similar assessment was performed using the predicted liver concentrations just based on the distribution of these molecules into the liver that was investigated in the non-clinical studies. Overall, despite of seeing some in vitro inhibition after all of the assessments, there was essentially no clinical DDI risk that was estimated for these molecules. As I mentioned, that also sometimes these drugs are to be administered on in addition to other treatments. So, for example, mipomersen is indicated for hyperlipidemia as an add-on therapy to statins, and then they looked at investigating potential for drug-drug interaction between mipomersen and statins, and also ezetimibe, which are the two treatments that patients with high lipid or LDL-C levels can be on continuously, and then you start a treatment with mipomersen as an add-on to help maintain the levels, the LDL-C levels low. Because these are continuous and common co-administered drugs, then investigating the DDI of these agents is a question that you likely received from the agency. Based on the in vitro data, there was no rationale to think of any potential DDI because mipomersen is not an in vitro inhibitor or does not interact with CYP450s. However, there's always the potential for a local PD effect as these drugs are acting in the liver and depend also on different biological mechanisms for the response and elimination, which can impact each other's drug or exposures. In the end, the overall conclusion here is that there was no impact of simvastatins or ezetimibe on the mipomersen exposures. However, mipomersen did reduce the Cmax that was observed for simvastatin and ezetimibe by about 50%, but that was not considered clinically relevant. Statins are drugs that have a high variability in the PK, and even if you look at the PK of patients, it can have really a wide range of concentrations observed. And at the end of the day, because of the long prolonged effect that we see with these agents, the assessments of PD response is important when you look at drug-drug interactions, where you may see a difference in exposures, but you may not see a difference in response. An interesting example is with givosiran. There was a drug-drug interaction with givosiran, which was not related to direct inhibition of the enzymes. So givosiran is an inhibitor of ALS1 mRNA, and ALS1 is the first and the rate-limiting enzyme in the heme biosynthesis. So if you decrease the hepatic heme, there is the reduction of the proteins that depend on heme, and that's the case for CYP450. So just based on the pharmacological cascade of events, there could be a reduction of proteins and impact then the CYP450 function. This was an interesting study from my perspective, a cocktail study that tested five major CYP450 enzymes in one study, and they investigated the impact of givosiran on 1A2, 2D6, 2C9, 3A4, and 2C19. The results demonstrate here on the figure on the forest plot that you have on the slides just show that there was a moderate inhibition of 1A2 and 2B6, and the increase in exposures of the sensitive substrates were in the 2 to 5-fold range when they were co-dosed with givosiran. A less moderate effect was observed with other enzymes, the 2C19 and the 3A, and no effect was observed with 2C9. The results of the study then had the label implications. So it's recommended that substrates that have a narrow therapeutic index or window for the 1A2 and 2D6 be administered with caution with givosiran. There has been no dose change. It's just a warning for these concomitant drugs. Switching gears to renal excretion now and contribution to the elimination of oligonucleotides. As Martin explained, there is accumulation of these drugs in the kidney, and they do undergo renal clearance. Usually GalNAc conjugated, they're less, you see less of a renal clearance for those type of molecules, but still it contributes to some part of the clearance of the parent. The approved drugs have investigated the impact in renal clearance mainly via a population PK analysis by enrolling patients that have different degrees of renal impairment into the clinical trials and investigating the impact or differences in PK or exposures for the drug across these different populations. There was no dedicated renal impairment study in the submissions except for inclisiran. Inclisiran was approved early in the year in 2020 in Europe, and a dedicated renal impairment study was reported for that compound. Interesting enough, if you look at the results for the renal impairment for inclisiran, so inclisiran is also GalNAc siRNA. It's indicated for the treatment of high cholesterol and high lipid on top as an add-on therapy to statins. The figure on the left just showed the PK profile of inclisiran in different groups, in groups with different renal function. The yellow curve on top is the severe renal impairment group, and the green curve on the bottom is a normal renal function group. So there was a clear impact of renal function on inclisiran exposure, which demonstrated the significant contribution of renal to the elimination of this compound. However, if you look at the figure on the left, this is the LDL-C levels by different renal functions. Essentially, there was no significant difference in the PD response in terms of reduction of LDL-C in the plasma across the different renal impairment groups. So despite of the higher exposures, the PD response actually did not change with the increase in the inclisiran exposures due to renal impairment. Investigations were also done in the clinical trials and looking at PD response as LDL-C reductions in patients that have different levels of renal function, and all of those demonstrated that there was no impact. So the end recommendation in the label was there was no dose adjustment necessary for patients with mild, moderate, or severe renal impairment and end-stage renal disease. From a oh, sorry, it's just from a very quick, as I understand here on the top of the hour, from a hepatic impairment perspective, also there has been no dedicated hepatic impairment study in a publication that reviewed the submissions for 5 different oligonucleotides. Some of this did do assessment of hepatic impairment impact on PK, doing population PK analysis. Again, for inclisiran, there was a dedicated hepatic impairment study. There was an impact of hepatic function on exposures, and there was an increase of about 2-fold in PK with increase in the degree of renal impairment, but the results on the PK and exposures did not impact the PD response. And the consequence in the label is that there was no dose adjustment that was required for inclisiran in hepatic impairment, despite of the differences of PK that were observed. So overall, in terms of the clinical pharmacology aspect, I just wanted to share a couple of examples and results of different studies and approaches that have been applied to the development of oligonucleotides. It is important that we leverage the knowledge that is available for the class, as well as the clinical pharmacology knowledge on how we do drug development to support understanding how intrinsic, extrinsic factors impact PK and what is the optimal dose regimen for patients. Regulatory agents are open and willing to work with the sponsors in developing oligonucleotides, so engage with them early they have guidances on how to engage with them in that aspect. So I think it's important to start a conversation as soon as you can. Key for this class, and maybe it's not usually not done for other molecules, is to also consider including PD assessments with your phase 1 studies, as changes in PK may not necessarily result in those adjustments just based on the PD response. And the FDA is also looking at publishing a draft guidance on clinical recommendations to support IND submissions of oligonucleotides. So there's more to come there. And with that, I just want to thank you for your attention, and we are going to open up for questions now.
Great, thank you so much, Verna and Martin. I'd like to encourage our audience to put their questions to our speakers in the questions box. Martin, someone wants to know whether they need IV data in monkeys to inform extrapolations if you intend to give your drug subcutaneously.
Yeah, that's a good question. I think personally I would say yes, and part of the reason for that is simply, you know, the difference or pitfall number one, where you have some multi-exponential decline systemically and then a mono-exponential decline, or a different profile subcutaneous than IV. So it becomes important to understand both aspects, and it becomes easier if you have a modeling framework, or you have a model where you can put all these different pieces of information together. You don't need a lot of IV data. You don't need to do every study with IV data, but having some idea, at least a pilot PK study with some IV data in monkeys would be highly beneficial to the rest of the exercise and the clinical development program down the road.
Great, thank you, Martin. Verna, someone wants to know, what's your thought on the need for assessing the potential for QT prolongation with oligos?
Yeah, I think that's an important point as well. You know, as I mentioned at the beginning of my talk, that there are other clinical pharmacologist aspects of the development of oligonucleotides that one needs to consider, and QTc assessment is one of them. A lot of these drugs that have been approved to the FDA or under development will run some in vitro assessments of QT prolongations or hERG assessments, and also do investigations in the clinical setting. So TheraQT studies have been done for these agents, as well as concentration QTc analysis. So far, what I've seen in terms of the publications of the compounds we worked with, and that there was no QT prolongation. But again, as I said, it's an evolving field. We are learning as we develop this drug, so I do recommend that we include the QT assessments during development.
Thanks, Verna. Someone wants you to explain, Martin, why oligonucleotides have such a long terminal half-life since they're highly soluble drugs.
So they have a long half-life because the pharmacodynamic effect, right? So they bind to risk. So it's all about the binding of these drugs to their target endpoint and all the interrelationships that implies. So because they bind to these profiles, they stick to the tissue, and they end up redistributing, and that ends up prolonging the half-life that you end up seeing.
Thank you. Looks like we've got one more question for you, Martin. Have you considered using PBPK modeling using preclinical data and IVIVE for first-in-human clinical studies, or at a later stage for drug-drug interaction evaluation?
That's a good question. I think there are some publications out there with some possible PBPK models that could be applied. We have to remember that PBPK models is an emerging field right now, so it's emerging across the entire industry, and it's certainly true of oligonucleotides as well. So I think we will see more of that in the future. And certainly I've seen some targeted use of these modules for certain particular molecules. So I do think in the future we're going to see more of that. Whether there's an actual framework in place, that's another question. But I think as the science evolves, we're going to see more and more of that, absolutely.
Okay, great. Looks like we don't have any more questions. Oh, sorry, looks like one more came in. I think this is for you also, Martin. Are radio-labeled QWBA studies done in rat? And what about radio-labeled animal, rat and dog metabolism studies to translate to humans, given the similarity across species?
I've seen some QWBA studies, and there have been some that have been done in multiple species. That's one of the basis how they hang their hats around its similar distribution across all species. So I've seen it. I've seen some of those studies in terms of QWBA and radio label studies to inform that. I think as the knowledge has developed, the need for those studies has decreased. And whether you need a particular one for your ASO probably depends on your particular program. But yeah, originally there were a lot of studies done in these species for these compounds and for QWBA. Yeah.
All right, wonderful. Thank you so much, Martin and Verna. And now we're going to pass it back to Pam to wrap things up.
All right, thanks everyone. Before concluding the webinar, we have a few announcements. Let's see if I can go to the next slide, please. You know what? I can just go ahead and just announce the webinar. The next webinar in the series will be March 17th, when Eva Berglund, Jackie Bims, and Lynn Georgopoulos will present expert tips for pediatric drug development success. You can register for all webinars by visiting Certara.com. On behalf of Certara, I would like to thank you for attending today's presentation. This concludes the webinar. Goodbye and have a great day.